questions about anything, you can go back and watch the recording if you want. Obviously, you can also reach out to me directly with any questions. But if you ever want to go through the notes again and kind of see how I talked about them, you can go back to this recording in order to kind of go through them once again on your all right, let me make sure we got everything loaded up here. All right. Yeah. All right, so just like with our past videos, once the video of our presentation tonight gets uploaded and it's ready to go, I'll post it here in the class notes video section of the Google Classroom. All right, so like I mentioned, we're going to jump into DNA, the structure of it, what are some of its different parts, and what is it doing inside of our bodies. All right, so DNA is an acronym for a big, fancy, scientific sounding word that we'll get to in a second. The cool thing about DNA is that there is DNA in every single living organism on the planet. So whether or not you are a bacteria cell, you are a type of plant, you are a type of bug, you are a deer, you are a human, every single living organism on planet Earth has DNA inside of it. And so it's shared by everything that's living. It has this kind of base DNA code. And the other cool thing about DNA is not only is it present in all the organisms in the world, it is also present inside every single cell of every organism. So the cell is like the tiny basic building block of our bodies. So you think like in humans, we have bone cells, we have muscle cells, we have nervous system cells. We have cells in our stomach, we have cells in our mouth, we have cells, our skin is made out of cells. Every single part of your body is made out of cells, and every single cell in your body has DNA inside of it. So, quick history lesson about DNA. For a long time, people knew that there was DNA in our cells, but they weren't quite sure what it was or what it looked like. They knew it had some different important parts on it that we'll talk about in a second, but they weren't quite sure how those parts exactly fit together to make this kind of helix structure that we now know as DNA. So the structure of DNA was actually first photographed by a scientist named Rosalind Franklin. And so she used a super fancy method of X-rays and took a whole bunch of pictures of DNA. And one of these pictures made this kind of iconic cross shape right here. So when she and other scientists saw that, it led them to the realization that this X pattern meant that there maybe were two different strands that kind of crisscrossed across each other, and that's what gave it this X. So for a long time, people thought it was like one big long strand that was kind of twisted up in a certain way. But it was thanks to Rosalind Franklin's picture that two other scientists named Watson and Crick were able to construct a 3D model of what DNA looked looked like. So they did this in 1953 and would eventually, in I think 1959 or 1960, they would win the Nobel Prize in medicine for determining the structure of DNA. So they put together this big kind of wiry structure, and they figured out that based on this kind of double strand that was wrapped around each other, it sort of looked like a spiral staircase. So if you've ever been in a house with a staircase that kind of winds up the wall or winds up the center of the house, that's sort of what the structure of DNA looks like. So DNA is an acronym that stands for deoxyribonucleic acid. Obviously, that is quite a mouthful, which is why pretty much everybody just refers to it as DNA these days. So we'll talk in a little bit later about why exactly it has this name. DNA, based on the Na part, is a nucleic acid. And so you can see we have these double strands here represented in sort of the gold yellow color that wrap around each other in this sort of spiral. And so that was the shape of DNA that Watson and Crick were able to figure out based on that photo from Rosalind Franklin. There are two strands that wrap around each other in sort of this spiral pattern. So DNA is a nucleic acid. And so if you remember, if you took biology A and learned about the macromolecules, nucleic acids are one type of macromolecules, and they are here to store and carry your genetic information. And so this genetic information, these genes, are all the information that goes into building and running the complex machinery that makes up you. So the instructions for how cells should be built, how they should be replicated, what jobs they should do, what color hair you develop, what color eyes that you have, your skin color. It's all based on your genes. So it's all based on this information that is stored in your DNA. So the DNA that's in every single organism on Earth and that's in every single cell of every organism contains the information to keep that organism alive and running. It's sort of like the blueprints or the user manual, the information manual for that organism and for the cells in that organism. So DNA is a nucleic acid, and it stores information about how to keep you and other living things up and running. So nucleic acids are not just two big long strings with all these little pieces in between, and that's it. They're actually made up of shorter pieces called nucleotides. And so we'll talk about these nucleotides more in a second. But basically, when you put thousands and thousands and thousands of these nucleotides together into a big long chain, you end up with your nucleic acids. So there's two different types of nucleic acids. There is DNA, which is the double-stranded type that we're going to be talking about today. There's also RNA. So instead of DNA, it's called RNA. RNA is just one single strand. And there are a couple other differences between RNA and DNA. but we'll talk about those in a later class. So the main thing to know here is that this DNA or this RNA is built out of thousands and thousands, if not millions, of these nucleotides all stacked up one after the other. And so we take this nucleotide, put a couple thousand or a couple million of them together, and we'll end up with a long strand of DNA or RNA. So just like how our DNA is sort of the blueprints for how our bodies work, the nucleotides are like the building blocks our DNA is based on. So speaking of nucleotides, every single nucleotide inside your DNA has three main parts that make it up. It has a phosphate group. It has what's called a pentose sugar, which, if you remember your elementary school shapes, a pentagram is a five-sided shape. So a pentose sugar is a sugar molecule made up of 5 bonds. So it's called a pentose sugar because it's a 5-sided sugar. And then the third part, and probably the most important part of the nucleotide, is the base, also known as a nitrogenous base. So there are 4 different bases that we'll talk about in just a second: adenine, thymine, guanine, and cytosine, usually abbreviated as A, T, G, and C. And so it could be any one of those 4 bases as our base group on the end here. So it's always going to be the same phosphate. It's always going to be the same sugar. And the nitrogenous base might differ between one of these 4 different ones. So every single nucleotide is made out of these 3 basic parts. And then if we put thousands of those nucleotides together, we get our DNA. So here, this one breaks it down a little bit more into some of the different elements that are involved. But we have our phosphate group. So that's what the P stands for here. It's phosphate. So we've got our phosphate group over here. We've got our 5-carbon or 5-sided sugar over here. And then it's connected to one of those 4 nitrogen bases. So 3 parts to a nucleotide, thousands or millions of nucleotides to a strand of DNA or RNA. All right, any questions about what DNA is, the shape of DNA, or nucleotides so far? Anybody have any questions about anything?
No, sir.
All right, thanks, Fernando. Anybody else? Any questions? Anything that you want to add over?
It's a thirst, immune eyesight, rash, itching, confusion, memory problems, muscle pain or weakness, joint pain, flushing, fever, or tingling.
No.
All righty. If you do think of any questions as we're going along, feel free to either ask them out loud, or you can also write them down in the chat. And I'll be keeping an eye on that as we go along tonight as well. All right, so we've got our 3 different nucleotide pieces. And this nitrogenous base on the end here is kind of the most important. Our sugars are, like I said, these 5-sided sugars made out of 5 different carbon molecules. So there's 4 carbons in this actual ring itself. that are kind of connected at the top by this oxygen up here. And then the 5th carbon up here is what connects it to that phosphate group. So you may notice that the 2 different sugar molecules pictured here look a little bit different. This one has OH coming off of this 2nd carbon over here. But this one only has an H. It's missing the O, the oxygen. So this is the ribose sugar, which is the one found in R and A. So that's where RNA gets its name from, is that it is a ribose nucleic acid. This sugar over here is missing the oxygen by this H down here. It's been deoxygenated, so it gets the name deoxyribose. So that is where the D in DNA comes from. It's missing that oxygen down here, so we say it's a deoxygenated ribose. So to shorten that a little bit, we just call it deoxyribose. So the D in deoxyribose is referring to the type of sugar that is in that nucleotide. So again, ribose, the RNA has ribose sugar. That's where the R comes from. DNA has deoxyribose sugar, which is where the D comes from. So the sugars are different between those two, but they don't really do a whole lot. They're just kind of the structure for the DNA or the RNA. The really important parts are our bases. So there are 4 different what are called nitrogenous bases, meaning that they're based on the element nitrogen. 4 nitrogenous bases in our DNA. We've got adenine, usually abbreviated as A, guanine, usually abbreviated as G, cytosine, which is usually abbreviated as C, and thymine, which is usually abbreviated as T. So these bases and these sugars and phosphates, again, we put thousands of them together to make our DNA strand. And so they link up with each other, one after the other, after the other, after the other. But remember, DNA is double-stranded. There's 2 different strands of nucleotides running by one another. So a molecule of DNA looks something like this. The sugars and the phosphates are on the kind of outside part of the DNA. Those are what make up the strands. And then the bases are connected to each other across the middle of the DNA. So if you think of a DNA molecule as sort of like a twisted up ladder, the sugar and phosphate form the kind of sides of the ladder, and then the bases form the rings going in between, or the rungs, I guess they're called. So these bases connect to each other in the middle of the DNA, and then the sugar and the phosphate run along the outside. So that's what's going on in this picture right here. This yellow part, this yellow kind of ribbon, is meant to represent the sugar and the phosphate. And then these little blue and yellow, green and red pieces right here, growing across the middle, are our bases. And they make these spirals on and on and on and on and on. So on each side has sugar and phosphate on the outside and bases in the middle. And then across from them is the same thing, sugar and phosphate on the outside, bases in the middle. So that is how the 2 different sides of the DNA, the 2 different strands, kind of get structured with each other. But it's not a completely random mishmash of which bases go together in the middle. So is there anything you guys notice about how the bases line up across from each other in the middle of our DNA structure right here? Is there any sort of pattern of how these bases are pairing up with one another?
A always goes with T and G always goes with C.
Exactly. Perfect, Pete. So yeah, Pete's pointing out that the A and the T always line up with each other. And the C and the G also always line up with each other. So this is something that is true for all DNA across all organisms. And it's called the base pairing rule or the base pairing law. So adenine and thymine, A and T, they always meet up with each other. So wherever you have an adenine on one side of the DNA, you'll have a thymine on the other side. And same thing flip-flopped. If there was a thymine on this side of the DNA, you'd have an adenine, an A, on the other side. Then same thing for guanine and cytosine, G and C. So if you have a G on one side of your DNA, then there has to be a C connected to it on the other side. If there is a C on one side of your DNA, then there has to be a G connected to it on the other side. For DNA, this is always, always, always true. There will always be T's and A's together, and there will always be C's and G's together. All right, so if you take nothing else away from the notes tonight, remember that A goes with T, G goes with C. So then again, we have that phosphate and sugar kind of siding going along the outside. So in this picture, they represent the dark blue is meant to represent the sugar part, and the light blue is supposed to represent kind of the in-between phosphate part. And then the A's and T's and G's and C's bond together across the middle of the DNA. Just, again, sort of like the steps on a ladder go across the middle. All right, so I want you guys to do a little bit of practice here with me. Let me make sure this is going to work the way I think it will. It does. So here is one side of our DNA molecule. And so based on this, I'm going to have you guys tell me what the other side of the DNA should look like. So, for example, on one side of the DNA, we have the bases A, T, A. So what is it going to look like on the other side of the DNA?
Is it TAT?
Boom, TAT. Number two. So because we have an A on one side, we have to have a T on the other side. Because we have a T on one side, we have to have an A on the other side. A on one side means we have to have a T on the other side. All All right, what about for our second group here? Somebody other than Noberto, tell me what is going to match up with TCA.
A.
Yeah.
Good.
IG.
AGT.
Exactly.
Yeah, IG.
AGT. So again, T on one side means that the A is going to be on the other side. C always matches up with G, and A always matches up with T. What about for our third group over here? What will match up with TGC?
A.
ACG.
CG.
ACG. Bingo. So T always matches up with A, G always matches up with C, and C always matches up with G. And then finally, what is going to match up with our final group of GGC?
CCG.
CCG. Bingo.
Yeah.
Sir.
Excellent. That is perfect. G on one side means that the other side is going to have our C. E matches with C, and C matches with G. So sometimes these are called, like our starting side is sometimes called the base side, and then, or the template side, and then the other side can be called the matching side or the complementary side. But that is exactly how the base pair rule works. If you know what one side of the DNA looks like, then you should be able to figure out what the other side of the DNA should look like as well. A's and T's always go with each other. So A on one side means you get a T. T on one side means you get an A on the other. G's and C's always go together. So a G or a C on one side means you end up with a C or a G on the other side. Very, very good. Excellent work. So the way that these bases match up, the way that they stay connected and hold the DNA together across the middle of our DNA ladder is through a type of bond called a hydrogen bond. So a bond is when two different atoms or multiple different atoms basically combine together. In the case of a hydrogen bond, this occurs when there's a hydrogen atom on one element, but there is another element kind of close by that it is attracted to. So, for example, here are the structures of some of our bases. And each one of these little circles, it's not written in because there'd be too many hydrogens and it would make everything messy. But each of these little circles has some hydrogens on them. And essentially, the hydrogens of one of our bases are attracted to the base on the other side. They don't fully go across. They don't fully get switched over from one to the other. But they form this kind of weak attraction between the two. So hydrogen bonds are, in general, fairly weak. It's almost like if you had two magnets that you held kind of close to each other, but not super close. So you could feel the pull a little bit, but it's pretty easy to break them apart. So that's similar to hydrogen bonds. So these hydrogen bonds are not very strong at all. They're very weakly attracting the two sides together. But remember, the reason why the DNA doesn't just kind of float apart from each other is that there are millions of hydrogen bonds in a single DNA molecule. All those millions of nucleotides that are stacked up on either side, they all have hydrogen bonds that are connecting them across the middle. So that is what the little lines in this picture right here are representing. So these hydrogen bonds are attracting the bases to each other down the middle of the DNA. And if there is just a handful of them like there are in this picture, then that might not be enough to keep it together and it might float apart. But DNA is millions of nucleotides long. And so there are millions of these hydrogen bonds that are all working together to keep the DNA molecule together as one big molecule overall. So essentially, even though they're weak, they are showing up in force. And this is what is helping connect these DNA bases to each other. So the reason that A's and T's are able to line up and C's and G's are able to line up is due to these hydrogen bonds between them. So the A and the T have sort of the right mixture of hydrogen bonds, and the C and the G have sort of the right mixture of hydrogen bonds to keep them connected to each other. So because we have all of these different bases in DNA, those all work like the letters of a code, essentially. So the different combinations of these millions of A, T, G, and C molecules determine what sort of traits that you're going to have. So one particular combination might mean that you have black hair instead of brown hair. Or one combination might mean that you have blue eyes instead of brown eyes. So like for this, I don't even know where this picture came from, this weird alien thingy down here. Maybe this species has a gene for hair color, kind of like we have genes that control our hair color. And so if you get a CAT gene, you end up with purple hair. Or if you just switch the C's and the T's, and so instead somebody else has the TAC gene for hair, they might end up with yellow hair. So the way that one of the ways I like to think of it is it's sort of like the letters of our language, right? There are 26 different letters in the English language. But with those 26 letters, we're able to create hundreds of thousands of different words, right? Just by combining those letters in different ways and different lengths. And we can put those words together in millions and billions of different sentences. So the basics of the DNA kind of work like letters of the alphabet. Sure, we only have 4 of them, but you can put them together in all sorts of different combinations in order to create essentially endless combinations of different types of the genes and different types of information. So even though everything on Earth has DNA as its sort of information storage, everything has different DNA depending on what sort of species that you are, right? So human DNA would look a little bit different from dog DNA, but those both would look a lot different from bird DNA. And those would look way different from fly DNA. And those would look even more different from bacteria DNA. So all these different species have the same type of DNA inside their cells, but they have completely different combinations of these bases. Again, there's millions of bases in the DNA of an organism. So we can get tons and tons of different combinations in order to create all of those different organisms that we see across planet Earth. Whoops. All right, any questions about DNA bases, DNA structure, hydrogen bonding? Any questions about anything that we have talked about so far this evening?
No.
All right, thanks.
No, mister. No, mister.
Thanks, Fernando. So I wanted to show you guys a little bit of a kind of preview of the homework that got posted today and tomorrow. Remember the attendance rule that since you guys are here in the Google Meet tonight, you do not have to do the Tuesday homework today if you want to wait and put it off for later. But it is really short and easy. So I would suggest going ahead and doing it sometime tonight just to get it over with. So the homework for tonight, the daily assignment for Tuesday, regards our friend the nucleotide right here. So if you remember, we talked a lot about the three different structures of the nucleotide. There's the base, there's the phosphate, and there's the sugar. What I want you to do is label in the spaces down here which of these parts of this nucleotide picture goes with the base, which part goes with the sugar, and which part goes with the phosphate. So again, if you want to do that on the Google form version, you're more than welcome to. There's also a Google Docs version, which is basically the same thing. So same picture and same labels. So like I said, pretty short and easy if you want to get it over with after we finish up class tonight. And then your Wednesday daily assignment has to do with matching DNA bases. So I feel pretty confident that you guys will be able to handle this pretty well since we just did it together and you got a great job a second ago. Let me see. Pete brings up a great point. So if you do, either you did the assignment already, or you're working on it later and you realize that you messed up the first time, you're always free to go back and correct your assignment if you made a mistake. And I usually will only look at the most recent one, the most current one that you turned in. So Pete, I'll make sure to look at the second assignment that you did. And that usually will be true for any of the assignments that I give you guys. If you do it the first time but realize that you messed up, you can go back in and do it again. And I will not hold the fact that you did it wrong the first time against you. I usually don't even look at the first submission. I just look at the second one. So that's a good point, Pete. Thank you for reminding me to bring that up. All right, so the Wednesday assignment that'll be going up tomorrow morning gives you a strand of DNA down here. And so I want you guys to write down what the matching strand would look like. So whatever matches with A, with T, with G, with C, with A, with C, and so on, that's what you'll write down for your answer below. This one might be a little bit easier actually on the Google Docs version, just because I was able to spread things out a little bit more and make the letters bigger, but it's the exact same strand, ATGCAC, TGG, ATC. And so I want you to tell me what would the matching strand of this DNA sequence look like? All right. Any questions on any of the homework assignments for today with the nucleotide or for tomorrow with the base pair matching of the DNA?
Nope. Everything's clean.
No.
No, thank you.
All right. Awesome. Sounds good, guys. So, I was going to take a look at something real quick. We'll save that for Thursday. So on Thursday, we're going to be talking about how DNA makes copies of itself. Hold on, where's that? There we go. So we'll be going over what exactly is the process for DNA going from this sort of tightly wound up structure to being unwound, broken apart a little bit, and all of the bases copied correctly into a new strand of DNA. It is a little bit tricky, and so I would definitely recommend looking at the notes for that one before we get started with class on Thursday evening. I'll do my best to explain it, and we'll hopefully be able to watch a couple videos on it, because actually seeing it getting replicated sort of in real time helps to kind of put all the pieces together. And so probably for Thursday, and then a little bit next Tuesday, I think we'll go back to it just because it is kind of a really complicated subject. And then the week after that, we'll get into how we use this DNA code to actually make the different proteins and structures that go into building up our cells and our bodies.